Our speaker for today is no other than Dr. Edwin R. Tadiosa. Dr. Edwin Tadiosa is Associate Professor 4 at the College of Science of Bulacan State University, a former Assistant Director at the Bulsus Research Management Office. He is now the Chair of the college's Environmental Science Program. He obtained his bachelor's degree from the University of the Philippines, Los Baños. He then acquired his MS and PhD biology degrees from the University of Santo Tomas. Dr. Tadiosa is a prolific researcher and educator in botany, biology, and mycology. He is involved in fungal research, which aims to determine all the macrofungi in the Philippines, particularly in declared protected areas. And with his accomplishments, Dr. Tadiosa has been recognized by different institutions and collegial bodies. For example, the Outstanding Asian Biodiversity Publication Award in 2012, given by the Liceo de Cagayan University, and the Outstanding Graduate School Research given by the Philippine Association for Graduate Research, Region 4A chapter. And the Japanese government and JICA has also given him a grant to visit several projects in 6 ASEAN countries, including Cambodia, Indonesia, Laos PDR, Malaysia, Thailand, and Vietnam. So without any further ado, everyone, let us all welcome Dr. Edwin Tadiosa. Sir Edwin?
Yeah, thank you, Sir Flor, for your kind introduction. So please allow me to share my slide. Okay, so again, thank you, Sir Flor, for your kind introduction. So good afternoon, everyone. At this point, I would like to thank Dr. Jennifer Nim. Wait, by the way, sir, I'm audible. Sir Flor?
Yes po, yes po. Yes.
Okay, sige. Okay, so good afternoon again, everyone. At this point, I would like to thank Dr. Jennifer Nim for inviting me to talk in this 2022 UPLB Museum of Natural History Biodiversity Seminar Series. And syempre, to her boss, her beautiful and talented boss, the UPLB MNH Director, Dr. Marian De Leon, and all the staff. So this afternoon, I will share to you, I will give you some informations about macrofungi across natural and artificial forest formations in the Philippines. The truth is, fungi are unpopular and less explored as compared to plants and animals or flora and fauna. But I would say this is equally important organisms simply because these are major decomposers in our ecosystem. Yes, ito po ay fungi. Hindi po ito Rafflesia. Ito po ay starfish stinkhorn fungus. In fact, one of the personnel in Region 7 in Cebu, Department of Environment and Natural Resources, contacted me and asking kung anong species ito daw ng Rafflesia. What I said that this picture, this fungus is stinkhorn fungus. Starfish stinkhorn fungus. Okay? Now, the question is, ano ba talaga yung fungi? What are fungi? Okay. What are fungi? Ano ba talaga yung fungi or yung fungus? The fungi are a large group of eukaryotic, spore-bearing, and achloropilus organisms, which constitute an abundant element of terrestrial biota in our country. Since this is a chlorophyllous organism, wala siyang chlorophyll. They are therefore unable to manufacture their own food. And as such, they have to obtain energy from the surroundings. Those which derive energy from dead organic remains called substratum are saprophytes or saprophytic fungus. And those which absorb nourishment from living tissues called host are parasites. So meaning, meron tayong saprophytic fungus and meron din parasitic fungus. And the fungi can classify as microfungi and macrofungi. Microfungi refers to group of fungi that form microscopic reproductive structures. And this include itong buni or Taenia flava, hadhad or Taenia croris, an or Taenia versicolor, alipunga or Taenia pedis. Ito yung usually na narinig natin sa commercial ng tusid. Ito po ay mga microfungi. And syempre, itong common na microfungi, itong molds or bread mold fungus. Ito yung mold sa tinapay. Rhizopus nigricans. And for the macrofungi, it refers to all fungi that produce visible protein bodies. And this include yung red cap fungus or Cookeina tricholoma, bird's nest fungus or Cyathus striatus, scarlet bracket fungus or yung Pycnoporus sanguineus, lacquered bracket fungus o yung Ganoderma lucidum, white jelly mushroom or Timelia possiformis. Parasol mushroom is another example of macrofungi. Ito yung Lepiota procera. And also the veiled lady mushroom or Dacryopora duplicata. Polyporus, Ganoderma, and Galliella rupa. According to Katibog, Sinha, and Heaney in 2006, our country is known to be one of the mega biodiversity countries. Numerous forest ecosystems within its tropical climate include mangrove forest, beech forest, Mojave forest, lowland rainforest, pine forest, and Mose mountain forest are present. The geographic isolation of the country and its ideal climatic conditions resulted in high endemicity among its flora and fauna. Though mycoploras, na kung saan yung fungi ay nakakategorize into this mycoflora, the mycoflora's endemism is not yet established. And since our country belongs to 17 mega biodiversity countries, our country is also listed as one of the most threatened ecosystem in the planet. This necessitates an urgent assessment of the country's biodiversity, including its poorly documented mycoflora. Many areas in the country remain unexplored or underexplored, and many species remain undiscovered in many of our unique habitats. According to DENR, UNEP in 1997, there are 4,968 species, fungal species. But there was no data on the endemic species. So may data lang dito ng endemic, angiosperm, gymnosperm, pteridophytes, bryophytes, so more on plants. Pero ang fungi, there was no data on the endemicity. And this 4,968 belong to 1,031 genera, or even more, resulted with the continuing research collaboration with foreign and local mycologists in the Philippines. Okay. All these fungi were deposited in all herbaria from Luzon, Visayas, Mindanao, though the major depository of fungal species in the country is in UP Los Baños and the Philippine National Herbarium. So ito po yung dalawa na herbaria sa Pilipinas na may pinakamaraming collections ng fungus sa Pilipinas. Now, the question is, saan ba ito nakita? Or saan ba ito makikita? Fungi grow almost everywhere, on land and in water. So kahit saan merong fungus. These fungi are found in diverse habitats from marine and freshwaters to various types of terrestrial habitats, including the mangrove forest to the lowland primary forest to the mossy mountain vegetation in the highest mountains in the country. Now, I was able to document some of these macrofungi in Luzon, Visayas, and Mindanao. Those protected areas and there's 3 major islands in our country. In Luzon, I visited and collected and documented fungus in Batanes Protected Landscape and Seascape, Northern Sierra Madre Natural Park in Cagayan, Aurora Memorial National Park, Catanduanes Watershed, Lipuon Point Reservation in Palawan, Mount Arayat National Park in Pampanga, Bataan National Park, Hinulugang Taktak Protected Landscape and Rizal. In Visayas, Rahasi-Katuna Protected Landscape and Bohol, Samar Island Natural Park, Sibalom Natural Park and Antique. In Mindanao, Mount Timpuong Hibok-Hibok Natural Monument and Camiguin, Mount Kitanglad Range Natural Park and Bukidnon, Mount Hamigitan Range and Wildlife Sanctuary in Davao Oriental, Agusan Mars and Wildlife Sanctuary in Agusan del Sur, Malagos Watershed in Davao del Sur, Initaw, Libertad Protected Landscape and Seascape, and Misamis Oriental. And to complete the documentation, I was able to explore other natural and artificial forest formation in the Philippines. And this include Basal Baobo Forest and Aurora, Dinagat Island Forest in Dinagat Island Province, Philippine Teak Forest in Batangas, Quezon Protected Landscape, Humunhun Island in Eastern Samar, Mount Manao San Cristobal in Laguna, Mount Makiling Forest Reserve also in the province of Laguna, Mount Macolot in Batangas, Mount Palay-Palay Mataas na Gulod Protected Landscape and Cavite, Busuan Peak and Bukidnon, Nueva Vizcaya Forest and Nueva Vizcaya, San Fernando Forest in La Union, Taal Volcano Protected Landscape in Batangas. Okay. In Aurora Memorial National Park, this park is one of the remaining wilderness areas in the country. The terrain of the park ranges from relatively low hills with dominantly moderate steep slopes near the coast to increasingly higher mountains with very steep slopes towards the central portion of Sierra Madre mountain range. AMNP is noted for its high species index of flora and fauna, including fungi. In Itaw Libertad protected landscape, the unique and exceptional beauty of the landscape is shaped by its harsh environment and the people who have learned to understand and live with it. ILPL hosts a number of rare and endemic species. Many of the species occur in small populations with limited distribution and are found in specialized habitat formations. Mount Panao San Cristobal protected landscape is largely viewed as a mystic mountain, with various religious groups believing in the healing powers of the mountains and its rivers. I will not forget this landscape since I reached the highest peak and discovered one of the fungal species of bird's nest fungus. Taal volcano protected landscape is small but terrible. Relatively small in nature, it is the second most active volcano in the Philippines. Local species and ecosystems are affected by volcanic hazards. Despite this and other threats, the rich biodiversity warranted its inclusion in 228 key biodiversity areas, or yung tinatawag natin na KBAs, and its classification as extremely high, urgent for biodiversity conservation. Quezon protected landscape is situated in the southern Sierra Madre mountain range on Luzon Island. The landscape is lowland rainforest with karst landscape and vegetation. The park is known for its virgin forest teeming with numerous wildlife, with several species endemic to the Philippines. Several species of macrofungi were collected in this landscape. Mount Kitanglad Natural Park, host of the few remaining intact rainforests in the Philippines. The park also provides sanctuary to an immense variety of flora and fauna, as well as several species of macrofungi. The indigenous communities have first and prior rights over the park's natural resources, as they have nurtured these resources over several generations. Academic researchers, some of you are connected or registered or enrolled in the university, some of the faculties. So you are considered as academic researchers. You need to seek permission from the Council of Elders to conduct studies in the park, and rituals are performed prior to that activity. But before entering those forest formations, those protected landscapes, those national parks, permit application is very important. If the application is approved prior to get into the forest and other areas to explore those amazing macrofungi, cortical will be done. Cortical will be done to inform the authority and the community of your activity in their areas of jurisdiction. So cortical in local government, cortical in the Department of the Environment and Natural Resources, and also cortical to barangay officials para at least alam nila kung ano yung gagawin nyo doon sa mga gusto nyong gawan ng study. But in protected areas, meron pang application na tinatawag na gratuitous permit. So you have to submit your proposal, your research proposal in the protected area management board to act on this proposal. And then again, if those body approve your research proposal, then ito yung mga pwede nyong gawin right after, before getting into your research areas. Okay. Exploring of macrofungi is not so easy. Hindi ganon kadali. Sometimes you have to traverse very wide and very deep rivers. You will hire people in the community to transport your equipment, supplies and materials, as well as your food and water using carabao or horse para madala doon sa inyong area. You have to establish a temporary shelter while doing your survey, collection, and photo documentation of fungal specimens. So, hindi ganon kadali, lalo na kung medyo malayo yung research study areas. Pero kung sa gilid-gilid lang, sa tabi-tabi lang, so hindi nyo kailangan yung ganitong klase ng arrangement in order for you to experience yung ganitong situation and condition to transport all your things in the study areas. Another question is, ano pa talaga ang importance of macrofungi survey? Bakit ba kinakailangan na mag-survey nitong fungi, nitong macrofungi? Why do we collect macrofungi? Una, for scientific research. Taxonomy, for ecological and biochemical analysis. Second, reference material for accurate identification of fungi. Kasi if you have already your collections and you deposit that collection in your school, in your laboratory, in the herbarium, and eventually mayroong proper identification, so magkakaroon ka na ng reference material to compare your new collection to that previous collection. And then reliable distribution data for taxa. Since you have already your data on hand, so at least magkakaroon na kayo ng idea, magkakaroon tayo ng idea kung ano yung mga fungus na meron doon sa mga specific areas. Like what I presented in my previous slides, collection from those natural and artificial forest formations in our country. And then fourth, information for production of fungal inventories for your local area. So, since sabi ko nga, hindi siya ganoon ka-popular as compared to flora and fauna or plants and animals, pero at least may mga initial study nang ginawa. So at least that data, that collection will become a part of the inventories for the different areas in our country. Okay. Normally, in field sampling, we use transect line and quadrat methods. Kung small yung area, at least 5 transect lines were established for each study site, na kung saan dapat strategically located. Parang hindi lang siya nakakompine sa isang area para at least ma-prevent yung sobrang biases, especially if your area is quite big. So dapat strategically located. So that transect line, you can establish, you can lay the 20 by 20 meter quadrat sampling with an interval of 50 meters of at least a length of 1,000 or 1 kilometer per transect. Sometimes opportunistic and purposive sampling are being used. Pero please take note, if you want to have a biodiversity or fungal diversity survey, kinakailangan na magkaroon kayo ng transect line as well as yung quadrat. Pero kung ang purpose nyo just to collect and collect and collect, so there's no need to just employ or apply opportunistic or purposive sampling. Kasi while walking along the trail, so pwede lang kayong pumolekta, pwede lang kayong kumuha ng sample ng fungal species. So that is opportunistic and purposive sampling. And if you have a bigger areas, so at least more transect lines, more quadrats, ang dapat gawin. Dapat i-establish. All fungal species found in its quadrat were identified, recorded, and documented. Simpson Index or Shannon-Weiner Index will be used in assessing fungal diversity in the different areas. But of course, you have to collect at least 3 to 4 samples. You can use that samples for future study, for proper identifications, and it will serve as herbarium specimens. So ito ngayon, yung magiging proof, ito yung magiging sample nyo na based on your data. So meron kayong support, meron kayong collections na magiging available just in case na mayroong mga researchers, scientists, faculty members, and students na gustong tingnan. Let's say, for example, if you have a study in Mont, so if those people are interested to see those collections in Mont, so at least merong sample specimens. Okay. Another question is, ano ba yung importance of measuring fungal diversity? Bakit pa hindi kailangan mag-collecta na lang ng sample? Bakit kailangan pang i-measure yung fungal diversity? Yung diversity ng fungi in a specific area. The diversity among organisms, including fungi, is necessary for the maintenance of the ecosystem. For the maintenance of the ecosystem. The more diverse the population is, the more the ecosystem becomes less vulnerable to perturbations. That is according to Groombridge in 1992. At the moment, biodiversity studies explore the possibility of fungi as indicator of environmental changes. Please take note, as indicator of environmental changes, aside from butterflies or moth, which are already accepted. Fungi are model organisms to work both in the laboratory and in the field, according to Hyde in 1997. They do not require elaborate trapping methods since they are just there. Nasa gilid-gilid lang, waiting to be collected and explored. The different fungal groups in their abundance on several strata from different habitat across the different geographical areas can be surveyed and compared. This may somehow assess whether the fungi fit to be good indicator of environmental change. That is the rapid destruction of the world's natural heritage. The methods used to inventory fungi are labor-intensive and require many years of collecting, according to Watling in 1995. Especially if you want to cover Luzon, Visayas, and Mindanao in our country. If you want to study the fungus, particularly the macrofungi in our country. Search may be done directly or indirectly on their associated host, either plant or animal, or even on other fungus. Kasi meron ding mga fungus na tumutubo sa fungus. So hindi lang fungus na tumutubo sa plants, hindi lang fungus na tumutubo sa animals, or even sa carabao dung and other kinds of dung. May mga fungus din na tumutubo sa fungus or restricted to a particular substratum. In order to assess diversity, it is essential to identify the species present. Actually, ito yung challenge. It's very easy to collect, but it's very difficult to identify, especially yung fungi. Okay. Some measures of fungal diversity includes species richness and relative abundance of the species. Species richness, it's the number or count of species in a sample or area. Relative abundance of the species, the more equally abundant the species in the area or ecosystem are, the more diverse it is considered to be. Okay. These are the representative macrofungal species, the ascomycetes, that I documented across forest formations in the country. And this include Cookeina solcipes, Daldinia, Galliela, Octospora, Pisaisa, Philipsia, Tarseta, Sailaria. And ito po yung mga sample pictures that I got while collecting and documenting those ascomycetus fungi in the different forest formations from Luzon, Visayas, and Mindanao. Sailaria, the carbon balls, the rubber cap fungus, Sarcosphaeria, the dead man's finger, or Sailaria polymorpha. So continue. Titingnan po natin, iba-iba yung color, iba-iba yung structures, and looking at those belongs to the different species of ascomycetus fungi. And another documentations, these are the representative macrofungal species. Ito yung mga Basidiomycetus fungi or Basidiomycetes that I documented. And this include Agaricus, Agrosaibe, Amauroderma, Armillaria, Astraeus, Auricularia, Boletus, Bisomerelius, Calocera, Cantarellus, Chlorophyllum, Clavaria, Clavolinopsis, Clethocybe, Polybia, Coniopora, Conocybe, Coprinillus, Coprinus, Coriolos, Corticium, Cortinarius, Crepidotus, Cyathus, Cymatoderma, Pleurotus, Polyporus, Porea, Saperilla, Psilocybe, Volkericium, Pycnoporus, Ramaria, Rosula, Schizopilum, Sterium, Stroparia, Termitomyces, Telepora, Termetis, Tremelia, Tricaptum, Tricholoma, and Bulbariella. So ito po ay ilan lang sa collections, ilan lang sa mga species that I collected and documented in natural and artificial forest formations in our country. And ito po yung pictures, sample of pictures that I took while doing the documentations. Ganoderma lucidum, Pycnoporus sanguineus, Microporus vermicipes, Microporus stabiliformis, Sclerotus striatus, Cymatoderma elegans, Tosola, Leotiltacta, Dacrymyces palmatus, Bulbariella, Bulbacea, Erlilia scabrosa, Dacryopinax spathularia, Auricularia auricula, Digistrum saccatum, or ito yung tinatawag na earth star, Ganoderma aplanatum, Schizopilum commune, Microporus xanthopus, Coprinillus disseminatus, Auricularia delicata, Tremelia pusiformis, Calbacia craneiformis. So again, just the same with the ascomycetes. So iba-iba din yung color, iba-iba yung shapes, yung structures, yung features. So by looking at the sample pictures, makikita agad natin na iba-ibang species ito. So if we have 4,968 species, so ito po ay ilan sa mga species na kasama doon sa figure na yon. In addition, okay. So, in addition, ito rin po ay makakasama doon sa publication. Actually, that is Ilshiber book publication entitled Mycology in the Tropics, first edition. So kasama po ito doon sa isang chapter nitong Mycology in the Tropics. And I think by November, baka magkaroon na ng copy or makomplete yung publication nitong book publication ng Ilshiber. Okay. So moving on, one of the highlights in the survey was the discovery of new bird's nest fungus at Mount Banahaw, San Cristobal Protected Landscape. Very, very small po ito. Kung makikita nyo po, actually yan po yung aking kuko. Kung i-compare niyo yung size nitong bird's nest fungus sa aking kuko, ay sobrang liit. Kung i-compare natin sa piso, itong bird's nest fungus na ito ay sobrang liit. Pero matured na po ito. So that is another species, a new species of bird's nest fungus. And when I viewed and look at the scanning electron microscope, ito po yung nakita kong features nitong bird's nest fungus na ito. Bird's nest fungus kasi kung titingnan nyo, para siyang nest, para siyang pugad na merong egg or itlog sa gitna. Kaya tinawag siyang bird's nest fungus. So ito po yung picture when I viewed and scanning electron microscope or SEM. And the description of this as well as the elevation. Nakita po namin ito sa 1,875 meters above sea level. Very close to the peak ng Mount Banahaw. And the species name is after the place where this bird's nest fungus was collected. It grows on a rotten branches, very small branch of agusip or Symplochus odoratissima, variety odoratissima, along the trail of mossy forest going to the peak. Kung ang peak ng Mount Banahaw ay 2,185, so very close na yung 1,975 meter above sea level ng Mount Banahaw, San Cristobal Protected Landscape. Before pandemic, the Philippine Postal Corporation recognized my collection and photo documentation by using my photos in featuring macrofungi and 2019 topical stamps issue. Gumamit po sila, humingi sila ng 10 pictures of species that I collected all over the country. At ito po yung 10 na species that I already mentioned in my previous slides. That is includes yung kabuting saging or Bulbariella, Bulbacea, earth star, or this stinkhorn fungus, and other sample. So 10 lang po ito yung ginamit nila in 2019. Para may feature in our stamps. And then, because of this documentation in our country, I was invited by the ASEAN Center for Biodiversity to join in the study fungi of Indonesia. This was supported by the East and Southeast Asia Biodiversity Information Initiative, the Ministry of the Environment, Government of Japan, ASEAN, and the Indonesian Institute of Sciences, or the Limbago Ilmo Pengetahuan Indonesia, or tinatawag na LIPI. And there's additional documentation while we are doing collection and documentations of fungi, particularly in Bogor, Indonesia. Okay. Molecular study is considered another trend in fungal study. Molecular identification and classification of species can be achieved using DNA barcoding. And this barcoding is a globally accepted method to confirm identity of organisms employing short genetic sequence obtained from a segment of genome. That is according to Herbert et al. In 2003. And according to Quinn et al. In 2012, the species can be identified quickly and precisely. Okay. The International Union for Conservation of Nature and Natural Resources, or IUCN, confirmed the need for fungal conservation by the establishment of a committee on fungi. A committee on fungi to stimulate activities worldwide. So worldwide, hindi lang sa Europe, hindi lang sa US, but all over the world, including Southeast Asia. The NIPAS Act, or the National Integrated Protected Area System, was introduced sa Pilipinas with the objective of developing a comprehensive protected area system and integrating the participation of local communities and protected areas management and decision making. Sa list dito, sa batas na ito, nakita yung importance ng local communities, yung participation ng communities in protection, in forest protection, and landscape protection, and also for decision making. The Act provides that the management, protection, sustainable development, and rehabilitation of protected areas shall be undertaken primarily to ensure the conservation of biological diversity, and that the use and enjoyment of protected areas must be consistent with that principle. The fungi cannot be conserved without the knowledge of the variety of species and their present distribution. There is a need to continue studies, particularly assessment of fungal diversity and study of changes in mycoflora over a period of time. In order to know how to conserve and protect. In order to know how to conserve and protect. To protect fungi is to protect mankind. Again, to protect fungi is to protect mankind. Nobody said that saving the hottest of the biodiversity hotspot is going to be easy. No single individual or organization is going to make the campaign to save the Philippine biodiversity hotspot a success. Partnership. Partnership is a key element. We have to be more inclusive. Doing it alone has not brought us far. The environment has deteriorated in the last 30 years. Biologists or mycologists alone cannot save biodiversity. Neither will biodiversity be saved without biologists or mycologists. Every species, every second, every hectare, in our fight to save the Philippine biodiversity, everything counts. The Department of Tourism, during the time of Secretary Richard Gordon, has a slogan, "It's more fun in the Philippines." Ito yung kanilang slogan, "It's more fun in the Philippines." In my case, it's more fungi in the Philippines. It's more fungi in the Philippines. And with that, thank you very much.
Okay, thank you very much, Sir Edwin. Especially for the stories that you have shared. There are so many things to discover in our protected areas, and the world of macrofungi is one of them. Thank you for enlightening us on some of the challenges that you have encountered in your research endeavors. Again, congratulations on your presentation, Sir Edwin. And
Thank you, Sir Flor.
And for our open forum, may I call on Dr. Jennifer Niem, our resident researcher at the museum's Mycological Herbarium, and currently our curator for macrofungi. Thank you, Jen, for moderating the open forum, and please take the floor.
Thank you, Kuya Flor. Again, thank you very much, Doc Edwin, for your talk. As you said, there's really not much study on Philippine macrofungi, and your talk somehow shed light on the current status of macrofungal study in the Philippines for the past 15 years. And I hope that this talk will enlighten people somehow from the audience that mushrooms and macrofungi are interesting species, and I hope more people will take interest in studying them. So anyways, as Kuya Flor mentioned earlier, I'm here to serve as moderator for Doc Edwin's talk. Again, to our participants, if you have questions, just type them in the chat box and I will read them for you. Alternatively, if you want to ask live questions, you may also do so. Just unmute your microphone and open your video. So our open forum is already open, and as there are no questions yet in the chat box, let me throw in the first question. Doc Edwin, how far do you think are we in the Philippines in unraveling the diversity of macrofungi or mushrooms that we have in our country? I mean, can you give us an estimate on how much of our macrofungi have been accounted for?
Yeah, thank you, Doc Jen, for your nice questions. So, unang-una, like what I've said, this organism is unpopular as compared to flora and fauna. So, in all study, in all assessment, laging flora and fauna, plants and animals. Pero hindi sinasama yung fungi. Hindi sinasama yung fungi. That is why ito din siguro yung reason kung bakit kaunti lang talaga yung mga studies na ginagawa dito sa pag-aaral ng macrofungi. And I would say siguro baka 15 to 20% pa lang yung estimate ko as compared to other study to other organisms. In fact, ang estimate number of fungus all over the world is 1.5 million species. That is estimate. And all over the world, there are 140,000 species recorded, documented, and described. 140,000 species. Pero ito, sa buong mundo. Kung pupunta tayo sa Pilipinas, mas kakonti. In fact, based on my presentation, there are only 4,000, more or less 4,000. And sobrang konti nun. And actually, yung 4,000 na yun, kasama doon yung microfungi and macrofungi. Ang macrofungi, more or less mga 1/3 lang. Actually, mas marami yung micro as compared to macro. So 1/3 lang siguro yung macrofungi kung i-compare natin doon sa bilang ng microfungi. So I would say napaka, in fact, behind tayo. Behind ang Pilipinas sa pag-aaral ng mushroom as compared to other country in especially in Southeast Asia. So maybe the University of Santo Tomas, University of the Philippines, De La Salle, and other SUCs nagsisimula. In fact, yung iba na una na like yung Big Four University, matagal na silang nagsimula na pag-aralan itong other organism na ito. But other school, universities, hindi pa ganon kadami yung mga studies. Kasi unang-una, ang challenge dito talaga is identification. It's very easy to collect. Sabi ko nga, it's very easy to collect, but it's very hard, very difficult to identify. And ito na ngayon, yung molecular. But bago magkaroon ng pag-aaral sa molecular study, una-una kailangan mag-undergo ng o pag-aaral tungkol sa molecular study. And second, dapat may laboratory. Kasi useless yung pag-aaral kung wala ka namang lab na gagamitin in your university. So yun yung mga actually nakikita ko na mga kinakailangan na gawin just in case na let's say yung isang university nag-decide na pag-aralan din itong another group of organisms na like what I've said, equally important in our ecosystem. Yes, Doc Jen. I hope I answered your question.
Yes po. Sir, i-follow up lang yung na-mention nyo nga yung difficulty in identifying macrofungi or yeah. So yung Nidula banahawensis nyo ba, have you sent your samples for genetic sequencing to ascertain that it really is a new species?
Actually, it's more analysis on so hindi na siya nag-undergo ng DNA analysis or molecular study kasi wala siyang katulad. So since wala siyang katulad, especially do sa group ng bird's nest fungus, so I think and we decided there's no need to have a molecular study. Kinakailangan kasi molecular study, especially kung may mga very close, especially do sa bird's nest fungus, may mga very close na collections, identifications. Actually, meron siyang real identity. Pero kung very close, so that's the time siguro na kinakailangan talaga ng molecular study. But since our collection ay kakaiba talaga siya, so we based our findings doon sa external features, doon sa morphological characteristics, as well as doon sa anatomical features or doon sa internal characteristics by using and by viewing at the scanning electron microscope.
But we don't know, we don't know if they have the same species abroad. Probably dito sa Pilipinas, wala. But for all we know, baka merong similar or the same species.
Yeah. That's why I explored other means, other herbaria, other publications, journals, books, and other materials in relation to that species identity. So wala naman. And also presented in scientific conference in order for them to know na mayroong ganitong klase ng discovery, particularly in the Philippines and Southeast Asia.
Thank you, Doc Edwin. We have a question from Darwin Ganado. And Darwin said, Doc Edwin, yun po bang tubig sa cup fungus ay pwedeng inumin? Thanks po.
Yung tubig, unang-una, yeah, Sir Darwin. Actually, Sir Darwin is connected with Bataan National Park. Pero gusto ko lang, yung tubig ay medyo, Ma'am Jen, medyo ano kasi yung audio mo, hindi ko ganong naintindihan.
Okay po. Kung pwede daw pong inumin yung tubig from the cup fungus.
Okay. So, actually, if there is water doon sa mga, especially doon sa mga ascomycetus, kasi yun yung mga cup fungus. So, normally, nagbibigay kami ng precaution. Kasi, especially kung itong mga species na to ay hindi pa ganong napapag-aralan, particularly yung chemical constituents. Kasi baka may
harmful microorganisms,
may features like spores. Baka medyo dahil hindi pa ganong napag-aralan yung chemical constituents. Baka may mga toxic or noxious substance na pwedeng i-emit ng particular species na yon. So yon. Pero in other areas, especially mga indigenous people, they do that. They did that for survival. Pero sabi nila wala namang masamang nangyayari. Pero that's case-to-case basis. Pero siyempre, kung kami tatanungin, so hindi namin ina-advise. Pero minsan, because of the experience, especially ng local people, especially sa place ni Sir Darwin, sa Bataan National Park, especially yung mga IPs doon, minsan iniinom nila. Pero yun nga, hindi namin ina-advise kasi baka may mga species. Kasi hindi lang naman sa isang species ng cap fungus or ascomycetes yung water present. Kasi kapag cap, siyempre, yun nakakapag-store yun ng water. Kaya yun yung take ko dyan sa concern ni Sir Darwin.
Thank you for your answer, Sir Doc Edwin. And then we have a comment and at the same time a question from Chabelita Avikivil. Good afternoon, sir. Thank you for the very interesting and informative talk. Is there an update regarding the diversity of macrofungi in Taal after its quite recent explosion? How were they affected?
Yes. Okay. Thank you, Chabelita. So, actually, sa tingin ko, wala after explosion. But the good thing is I visited that area, particularly sa Talisay Park. I documented at least 98 species of macrofungi and Taal Volcano, Talisay Park, and also dito sa Taal Volcano, Cuenca Park, near Mount Makulot. Pero after explosion, I think walang nagawang pag-aaral o study with regards to collection and documentation of this particular organism. And kung siguro other researchers, students, faculty members, kung pag-aaral particularly yung malapit like Batangas State University, ito yung pinakamalapit na university dito sa area na to. So mas maganda na mapag-aralan nila. So in fact, I have one collaborator in Batangas State U with regards to this documentation. So siguro mas maganda kung itong faculty na ito ay makabalik at malaman kung meron bang effect yung explosion ng Taal Volcano doon sa existing ng fungus doon sa particular area, doon sa Taal, doon sa paikot ng Taal. Kasi ang Taal Volcano is situated, napapaikutan ito ng 12 municipalities ng Batangas. So mas maganda na magkaroon ng study tungkol dito. Para at least yung effect, malaman yung effect, and malaman kung it could really affect sa growth and development of fungus doon sa Taal Volcano Protected Landscape Area.
Okay, thank you.
Yes po, Ma'am Chabelita. So wala po.
Okay, and then we have another question from Justin Gabriel Badis. Good day, sir. With regards to fungal diversity, ano po status natin with regards to invasive species of macrofungi, especially sa agricultural sector? Do we have species of fungi that we should be worried about, especially those that may displace or compete with other native endemic fungi? Wala
Okay,
pa naman
actually, Sir Justin, with regards to fungal diversity, wala pang study, wala pang pag-aaral ng invasive species, particularly sa macrofungi. Wala pang nagawang study, even yung research institution like BPI, under of the Department of Agriculture. Ito kasi yung institution na talagang ang focus is invasive species. in addition dito sa mga state universities. So wala pang nagawang pag-aaral tungkol dito sa diversity, especially macrofungi, invasive species of macrofungi, particularly sa agriculture. Kaya I would say in terms of yung presence ng macrofungi sa farms, agricultural farms, medyo safe pa. Wala pang invasive na kami nakikita at wala pang pag-aaralan at wala pang observations na yung presence nitong particular species na to ay nakakaapekto mismo doon sa plants, native plants. Unlike yung mga invasive plants, invasive animals, kita agad natin na even from a distance, nakikita natin yung effect nitong plants and animals, itong invasive plants and animals, doon sa existing na native and indigenous species. So wala pa po dito na pag-aaral about sa invasive species ng macrofungi. And hindi pa tayo kinakailangan na mag-worry. In fact, napaka bilis nitong especially mga fleshy na minsan oras lang or minsan 1 day or 2 days lang bulok na. Natuyo na yung fungus. Pero yung mga woody fungus, mga wood-rotting fungi, ito yung tumatagal. Pero ang target naman nito, yung substrate nito, yung mga rotten wood, rotten bamboo, rotten and other kinds or types of rotten materials. Ito yung pinaka-substrate o substratum nitong macrofungi nito. Kaya may tinatawag tayo na saprophytic fungus. Kasi ang target nito is yung mga rotten materials or rotten wood. And yeah, so wala pang nadi-displace na in fact sa ngayon napakahirap ding i-distinguish yung pagiging native or indigenous ng species ng fungi. Kasi po ang fungi ay cosmopolitan. Napaka-wide ng distribution. So yung mga species natin, makikita din siya in Southeast Asia up to Australia. Yung species natin dito ay meron din sa mga bansa na yon. Kaya napakahirap pang establish yung macrofungi na native and indigenous. Although may mga microfungi na nasa part ng database na considered as native by looking at the specific epithet. Pero mga microfungi po ito. Yes, I hope I answer Sir Justin your query on invasive species. Ma'am Jen, naka-mute ka po.
Good afternoon, sir. Question from Gazelle Rule. As to your expertise in macrofungi in mostly ICCA areas, do you also conduct ethnomedicinal studies related to macrofungi?
Yes. Actually, although not directly, because I have some collaborators. But to address your concern about ethnomedicinal, meron akong isang collaborator from Nueva Ecija. So we studied the species of plants, especially used by the natives and Nueva Ecija and Nueva Vizcaya. So we have data already, and one or two faculty members in Central Luzon State University, we're also conducting ethnomedicinal studies in Central Luzon areas. Kaya dito, napaka-importante nung collaborations ng mga researchers, ng experts, faculty members, or even students para at least magkaroon ng mas maganda and mas wide in scope, mas malawak yung pag-aaral, hindi lamang sa taxonomy, but also in this particular yung ethnomedicinal properties ng macrofungi. So that is another study, ethnomedicinal studies nitong macrofungi na ito.
Thank you, Sir Edwin. So we have question from Florante Cruz, our extension coordinator at UPLB MNH. Hello, sir. Out of curiosity, may I know your perspective on macrofungi collection vis-a-vis the gratuitous permit in light past issues concerning people collecting not the actual specimen, but reproductive parts, which they can rear in the lab, thus bypassing the GP policies in terms of collection and transport of macrofungi.
So it's about the GP. It's about the gratuitous permit. And yeah, concerning people collecting not the actual specimen but reproductive parts. But actually in Wildlife Act, naka-stipulate doon na even yung derivatives. So ibig sabihin, hindi lang yung actual specimens, hindi lang yung actual fungus, hindi lang yung actual sample, kundi pati yung derivatives nito ay ipinagbabawal din ng batas through that Wildlife Act. Kinakailangan din na humingi ng permit sa sa Protected Area Management Board, particularly kung protected areas. So if that area is not protected, so kinakailangan ding humingi ng GP or PIC or prior informed consent sa local government. So dalawa ito, kung protected areas sa Protected Area Management Board, kung non-protected areas, kailangan ng prior informed consent sa local government kahit na hindi siya talaga yung actual specimens, kahit yung pinaka-reproductive parts lang. And yeah, actually maraming cases na ganito na hindi lang sa fungus, hindi lang sa macrofungi, but also in plants. Na kumuha lang sila ng seeds, kumuha lang sila ng fruits. Sabi nila, seeds lang naman po yan, fruits lang naman po yan. Pero sa batas, kinakailangan na mag-undergo ng collection permit process dito sa mga authority na ito. Kaya kahit pa siya reproductive part, although kung reproductive part, sabi natin, madali naman siyang i-secure. Meron lang tayong bottle, small bottles, plastic, bags, and other materials, madali siyang i-secure. Pero sa batas ay bawal pa rin. Kaya yun nga, kasi nga parang gano'n na dahil naman parts lang siya, parang madali siyang i-secure. Pero again, nababypass yung policy or yung provision doon sa Wildlife Act patungkol sa collection of organisms including their derivatives. And yung reproductive parts kasi considered siya as derivative. I think that is also being part of that particular Wildlife Act. Kaya ganun din actually sa transportation. If you want to transport, say for example, from Baler, Aurora, going to Los Baños, Laguna, hihingi ka din ng transport permit from the Department of Environment and Natural Resources to transport your collection. Pero ang nangyayari, dahil nga parts lang siya, or even kahit itong macrofungi, dahil hindi siya bulky, pwede mo siyang ilagay sa bag, At may transport mo siya, makakasakay ka ng Genesis, makakasakay ka ng bus, going to Manila, and going to Los Baños. Pero the thing is, na-violate mo pa rin. Although hindi ka lang nahuli ng proper authority, hindi ka nahuli, pero just the same, you violate the law for that particular aspect. Kaya lang, yun nga, minsan may mga nakakakita, so i-inform yung authority like the DENR na merong ganitong tao na they collected this particular sample specimens, and yun lang yung nahuhuli. Yun lang yung na i-intercept ng authority. Pero if there's no such kind of observations, so lusot na. Madadala sa laboratory. So that's my take on that. I hope Sir Flor, I answer your query or questions with regards to gratuitous permit, particularly for the specimen reproductive parts.
Okay, this should be our last question. Again, from Chabilita Avikivil. Hello again, sir. We are aware of how fast nature is being destroyed, especially because of climate change and destructive human activities. Is it really possible to know the extinction rate or being threatened of Philippine macrofungi species, or further studies still need to be done?
So, Ma'am Chabilita, thanks again for your another question with regards to the extinction rate or being threatened. So, dahil kaunti lang yung nagkakandak ng study, particularly in this group of macrofungi, hindi natin malaman yung unang-una kung ano ba yung species ng macrofungi yung nawawala. At kung may nawawala, ano yung rate ng pagkawala nito because of so many factors, including climate change. Kaya I encourage everyone, especially yung nasa loob ng Zoom, if you are connected with the university as faculty members, researchers, or even the students, please try to venture or mag-aral dito sa group na ito. Para at least mabuo natin, magkaroon ng data from Batanes up to Hulo. Magkaroon ng data by region, by province, by cities, and by municipalities, and sana by barangay. And by doing that, although napakahirap gawin, it takes time. Sabi ni Watling, it takes time. You need so many years to do that. Pero kailangan nating mag-start, kailangan natin magsimula. And by doing that, doon natin lang malalaman, Ma'am Chabilita, by looking at the data, kung mayroon bang nawawala, mayroon bang nagiging endangered, nagiging extinct. Di ba sa plants, meron tayong status, meron tayong conservation status, meron tayong endangered species, meron tayong extinct, meron tayong least concern. Pero yung sa plants kasi, established na sila na kapag endangered na siya, meaning candidate na for extinction. So dahil candidate na for extinction, maybe several or kaunti na lang number of individuals yung present. Yung makikita and in artificial and natural forest formation. Pero without that kind of study, napakahirap malaman kung mayroon na bang nawawalang species. Eh syempre, mas mahirap lalo malaman yung rate of extinction kung wala tayong data ng macrofungi all over the Philippines.
Okay.
So I hope, Ma'am Chabilita, I answer your query, your questions.
Yes. So thank you, Doc
Please
Edwin.
join us in this endeavor.
Yeah. So thank you, Doc Edwin, for graciously answering the questions, and to all the audience for actively participating in the open forum. Now I will turn you over to our MC since we don't have any more time. Take it away, Kuya Floor.
Yes, thank you, Ma'am Jen. Thank you for moderating. And to Sir Edwin, congratulations on your presentation. So we'll
Thank
be
you po.
wrapping up our program. Let me just share my slide here. We will be awarding this certificate of appreciation to our speaker, Dr. Edwin R. Tadiosa, and it reads, the University of the Philippines Los Baños, UPLB Museum of Natural History is awarding this certificate of appreciation to Dr. Edwin R. Tadiosa for serving as our resource person during the UPLB Museum of Natural History Biodiversity Seminar on exploring amazing macrofungi across natural and artificial forest formations in the Philippines, held today, August 3, 2022, from 2:30 to 4 p.m. via Zoom. Signed by our director, Dr. Marian P. De Leon. Congratulations, Sir Edwin, and
Okay, thank you, Sir
splendid talk,
Flor.
sir.
Thank you, Doc Jen, and please extend my regards to Director Marian De Leon and all staff of the UPLB Museum of Natural History. Again,
Thank,
also thank you, and have a nice day.
thank you, sir. Will do.
Yeah, our pleasure, sir.
We invite you to look at our website. It's at mnh.uplb.edu.ph, or if you have questions or some concerns, do write us at mnh.uplb@up.edu.ph. We are present in social media. We are on Facebook, Twitter, YouTube, and Instagram. Please like and follow or subscribe. Just search for our handle UPLB Museum, and check out our articles at Wikipedia and TripAdvisor.com. Our next webinar will be on August 22, and we will have an orchid breeder talking about biodiversity and breeding practices um, of orchids in Hawaii. Just wait for our public service announcement via Facebook. And with that, maraming salamat po sa inyong lahat, and have a great day, everyone.